So my name's Edel O'Toole. I'm an academic dermatologist in London, and I'm pleased to welcome you to this week's ESDR Kitchen Live webinar. For those of you who are new to the webinar, these are live events in lieu of being able to meet face to face, and so far have been very successful. There are 4 formats. There is the recipe book where new research techniques are explained in the ESDR kitchen. There is sweet and sour where 2 academics debate a hot topic. There's freshly baked, which we have today, where the latest high impact research findings are discussed. And molecular cuisine, where research leaders describe their discovery pathways and how they got into skin research. This CSDR Kitchen episode is kindly sponsored by Amgen and Sanofi, and we are very grateful to them both. If anybody has any questions, we'll have questions at the end, and if you could please type your questions in the chat. So there are 2 speakers today. The first speaker is Sally Dabelsteen, who I'm very pleased to introduce. She is an associate professor at the University of Copenhagen in Denmark. She is a dentist by training and has also a PhD, which she obtained in Copenhagen. She has done a lot of work on epithelial mesenchymal interactions and generating 3D models. And she's here today to talk about her recent first author paper in Developmental Cell, dissecting the role of glycans in epithelium using CRISPR-Cas9 technology. So Sally, when you're ready, you can share your screen and start your presentation. Thanks.
Thank you very much. So thank you very much for letting me present my study with dissecting functions of glycans in human organotypic models that I did together with Emil Pallesen as a co-first author. So the glycans are involved in the vast majority of biological functions, involved in our blood type antigens, pathogen infections, the mucus protection of our cells in the immune systems, and in protein folding. And glycans come in various models, and they are made by more than 200 glycosyl transferases, important for the initiation, branching, and elongation and capping of glycans. So how do we dissect the function of these glycans? Because one enzyme can put different glycans on many proteins that are in a very complex manner. So we used a genetic approach to dissect the function. And as you can see, glycans are comes in various modifications. So we have glycosphingolipids, proteoglycans, oligoglycans, and N-glycans. In our study, we decided to knock out different enzymes involved in the elongation of these glycans. We used CRISPR-Cas9 delivered with the lentivirus to immortalize the keratinocyte cell line from Jim Reynolds' lab, the N31, which makes a nice organotypic model with a nice stratified epithelia. So we use these knockout cells to make organotypic models in order to deconvolute endogenous effects of individual glycogenes on tissue formation. So to start with something that many of you probably know, the Notch pathway. So Notch is actually glycosylated, it's O-glycosylated, and also its ligands, jagged and delta, are glycosylated, by two enzymes called POGLUT and POFUT. They do this in the, this is done in the Golgi apparatus. And as you can see, it is important for the binding of Notch to its ligands. And you know that Notch is important for the differentiation of epidermal, for the differentiation of the epidermis. So to validate our model, we decided to first knock out Notch 1 to see if it fits with the results found in mice. And knocking out Notch 1, we saw a enlarged area of basal cell compartment, a delayed differentiation, which has been shown before in Notch 1 knockout mice. Also, knocking out the ligand JAK1, we saw a enlarged basal cell compartment and a complete absence of the spinous layer, which is also has been seen in mice. So this actually fits very well with the results seen by other researchers before. Then we said we can use our model. We knocked out the POFUT1, for instance, which is the enzyme that adds the fucose on jagged in order to get jagged to bind to Notch. And knocking out this enzyme, we saw a lack of spinous layer, as we saw in the jagged knockout, defining the function of fucose in the modification of Notch signaling. Also with the knocking out the POGLUT, which adds the glucose, we saw a delayed differentiation and a lack of inhibition of P63, which fits very nicely with the inhibition of Notch function, and thereby finding distinct functions of both POFUT and POGLUT in human tissue formation. So going to N-glycans. So this is a special phenotype. You see knocking out complex N-glycans, which are really important for protein folding. We have a phenotype with knocking out the initial enzyme NGAT1 with large vacuoles, even though the cells still form a stratified epithelium. Knocking out the NGAT5, which is elongating these end-linked glycans, complex end-linked glycans to tri-antennary glycans, we see a more subtle phenotype. So these vacuoles, what are in these vacuoles? We decided to look with the electron microscopy, and we found that they are filled up with small vesicles resembling lamellar bodies, which are present in the differentiated layers in the human skin. And we speculated that this could be due to loss of control of the specialized transport in these cells. And here we saw also that TGN46 with trans-Golgi network marker and the early endosomes are almost absent in these cells. Also, we have a complete lack of filaggrin, which also should be is transported by vesicles. So this really links the complex N-glycans to a transportation defect if these are abolished. So it's been speculated before that in cancer that N-glycans, complex N-glycans are involved in cadherins and desmogleins. But in our study, the previous studies have been done with point mutation, where you completely abolish ending glycans. And as I said before, they are very important for protein folding. So in our study, we saw no effect on cell-cell adhesion, either with the dissociation assay, so functionally, and also a nice display of E-cadherin and desmogleins, and we have nice desmosomes forming in this tissue. So complex N-glycans are as such in these cells, not involved in cell-cell adhesion. Then we tried to look at in a more disease-like phenotype, so making a wound in the organotypic model. And here we saw a specific phenotype with the MGAT1, where the cells fail to heal the wound and stop migrating. So we thought, what could this be? And going as a logical way would be to look at the integrins, and almost all integrins actually are displayed normally in all our knockout cells, except for integrin alpha 5, which has also been shown to be linked to wound healing. And we see in integrin alpha 5 being localized inside the cells in the MGAT1 knockout and not transported to the membrane. So, looking at the glycosphingolipids are involved in the formation of stratum corneum and also controlling tyrosine kinases. And it's been widely studied in mouse studies where most of the knockout mice have been focusing on the initial enzyme, the glucose synthase, which puts the first glucose on a ceramide. So when you stop these glycosphingolipids, you will have an accumulation of ceramide, and in our study, we see accumulation of apoptotic cells, possibly due to this accumulation of ceramides. So apoptotic cells and also with tunnel assay. So, but in the later chains of glycosphingolipids, we see a loss of basal cell integrity and an decreased conification in the initial glycosphingolipid knockout, which fits with the mouse phenotypes that where knockout mice die early due to permeability defects. So in our model, we did not see any permeability defect from the top of the skin, but adding dye from the bottom, from the basal compartment, we see a massive defect in barrier, which also fits with the EM pictures, where we see fewer desmosomes and fewer tight junctions. So going to the tyrosine kinases, which has been shown to be regulated by glycosphingolipids, we also in our study saw that phospho-IGFR and phospho-EGFR are highly upregulated in glycosphingolipid knockout cells. And possibly those are the ones that affect the basal cell integrity. So lastly, just a little word about the O-glycans. So the O-glycoproteome has been widely enlarged during the last 10 years, and we have identified more than 10,000 sites of possibly O-glycoprotein sites in different proteins. So galnic O-glycosylation has been shown to be linked to cancer. And we have in our previous study showed that P53 mutated epithelial cells will actually, if having truncated short O-link glycans, become invasive in mice. In these normal, not mutated epidermal keratinocytes, we see only loss of cell-cell adhesion, so the cells will dissociate very fast, and we see a delayed differentiation. And this fits very nicely with our findings in the possible O-glycosides, which has been shown to be on integrins, CD44, laminins, and cadherins, for example, fitting with our cell-cell adhesion problem in these cells. So to summarize, this genetic engineering tissue, we can define distinct phenotypes associated with the loss of individual glycosylation pathways. With N-glycans, we see effect on lamellar bodies and wound healing, organotypic on cell-cell adhesion, and glycosphingolipids affect epithelial integrity and barrier formation. And the O-fucose and O-glucose differentially regulates notch signaling. So we hope this platform will help defining the glycome in epithelial homeostasis, transformation, and host pathogen interaction. Thank you very much.
Thanks very much, Sally. So any questions for Sally, please put in the chat, or questions and answers. So we move on to the next paper. So the next speaker is Dr. Gary Reynolds, who's a rheumatologist in Newcastle in the UK. He was a Wellcome Training Fellow and obtained his PhD in 2016. And since 2018, he's been a Wellcome Career Development Fellow, which is a very prestigious award in the UK. His main interest is in the pathogenesis of vasculitis, and his current Wellcome grant, I think, is to look at the pathogenesis of giant cell arteritis. But on the way, he's spent some time in the laboratory of Ms. Hanifa, where he has produced this amazing work with a large group of people on single cell RNA sequencing in skin, in atopic eczema and psoriasis. So this is something which will be of interest, I think, to every skin scientist. And it was published in Science in January. So Gary, we look forward to your talk. Thanks.
Thank you very much for the introduction. Yeah, thank you. That's very kind. And thank you for inviting me to talk today. So as Edel said, this is the work of a large group of people I will acknowledge at the end. We've looked at changes in the skin between development in healthy skin and then again in disease states. We've looked at atopic dermatitis and psoriasis. So the skin has a broad immunological and physical barrier function. So physical through the keratinocyte layer and the tight junctions, and immunological through the dense and complex immune network that's present in skin. But in development, skin is only a couple of cells thick, which is fine for the sterile aquatic environment in utero, but it needs to prepare for the terrestrial and very much non-sterile environment we face from birth. And so it undergoes significant changes. So for example, in the dermis, there's vasculogenesis, so a vascular network to support the developing structures, and recruitment of immune cells to prepare ourselves for the challenges we'd face from birth. And this brings us to healthy skin, but healthy skin, both of these processes can go wrong. So eczema and psoriasis is no two common inflammatory skin diseases, both having features of aberrant vasculogenesis and immune cell recruitment. So we performed a comparative analysis across development, health and psoriasis. So the N number here is the number of donors, and the K, the number of cells that we analysed. So the tool we used to perform this was single cell RNA sequencing, which is becoming increasingly common. So here we looked at the transcriptome of single cells. Using this, we can classify the cells. This UMAP here is a two-dimensional representation. Each dot is a single cell and each color is a distinct population. We can look at what the cell's producing to classify them. So we find cases keratinocytes in tissue as we'd expect. We find melanocytes. We find all of the populations we'd expect, but with some perhaps unexpected heterogeneity. So B are the fibroblasts, the V are the vascular endothelium. The vascular endothelium, we find different populations of vascular endothelium in the skin. In the APC compartment, we find different types of macrophages. So we've just called them, just numbered them MAC1, MAC2 for simplicity's sake. But there's an unexpected heterogeneity in healthy adult skin. So first, we formed a quantitative analysis across stages, and the message here really is the increasing complexity as we go from development to adulthood. So in the lymphocyte compartment in development, it's almost exclusively NK cells and ILCs, but we can see in the top right, when we reach adult skin, there's predominantly T cells, and this complex different types of T cells as well. In development, in the myeloid compartment, it's predominantly macrophages, but then when we reach healthy adult skin, there's Langerhans cells and different types of dendritic cells. And so perhaps some of the complexity isn't really captured in these bar graphs. In the stromal compartment, it's almost exclusively fibroblasts in developing skin, but we can see pericytes, endothelial cells, Schwann cells, melanocytes appearing in adulthood. So quantitatively, the change from stages is complexity. Then coming back to this heterogeneity that I mentioned before. So when we look at macrophages, we see two populations marked by, so they have macrophage markers, but in addition, they express some additional markers to distinguish them. So NR4A1 and 2 are transcription factors that we find exclusively expressed in this MAC2 population. In the vascular endothelium, we find three populations, in particular this vascular endothelium 3 that's marked by SOX3 and this atypical chemokine receptor ACKR1. So we can look at what different markers these populations express to distinguish them. But what does it mean? So it's not initially clear, but what we did note on the left here is that this MAC2 and vascular endothelium 3 population are both expanded in disease states. So at the bottom, we've got healthy adult AD, atopic dermatitis and psoriasis. So we can see expansion of MAC2 in disease and expansion of vascular endothelium V3 in disease states. Because of the cost and complexity of performing these experiments, we have the N numbers aren't big, so we can go back to look at bulk RNA sequencing data and use a process of deconvolution to try to make the predictions more broadly. So here we've used data as below of around 50 donors each from atopic dermatitis and psoriasis, and again there's a prediction from deconvolution that there's an expansion of MAC2 and V3 in the bulk lesional psoriasis and atopic dermatitis. So when we've found this population healthy adult skin, we found it's expanded in disease states. How does this link with development? Well, interestingly, when we align our developing and adult skin populations, mostly we find what we would expect to find. So mast cells in developing skin align with mast cells in healthy adult skin. But interestingly, the developing macrophages align with this disease-enriched MAC2 population. Similarly, in the non-immune compartment, we find what we'd expect to find, but in the vascular endothelium, the vascular endothelial cells from developing skin align most closely with vascular endothelium 3, this disease-associated state. We can expand that and extend that finding to other data sets. So there are people are looking at developing tissues from gut, kidney, liver and thymus. And again, the developing skin macrophages and endothelium align with this V3 population and MAC2 population. So there seems to be a cross development. What does this all mean? So we sought to validate this MAC2 phenotype. So F13A is a marker that picks out this disease-associated MAC2 population. So we performed microscopy of disease skin and found that there was an expansion, or there was an increase in this MAC2 population in atopic dermatitis and psoriasis. Furthermore, following treatment with methotrexate, this population was decreased. But what's it doing? So to try to answer that question, we looked for conserved programs, so genes that are expressed in the developing macrophage population and in the disease MAC2 populations in eczema and psoriasis. So with this enrichment analysis, we find that the top program that comes up relates to vasculogenesis and VEGF production, so upregulation of VEGF. We do the same thing with the vascular endothelium. So what are the vascular endothelial cells doing? So we look at the conserved genes both from development and in diseased skin. And of the programs that come up, there's repeatedly references to immune cell recruitment and chemotaxis. We stain for these cells. So on the left here, we use PCAM1 as a pan-vascular endothelial marker, and SCGN is gamma-synuclein, so that's specific for VE3. And we find co-localization of the gamma-synuclein with the more dilated vascular endothelial cells, or the more dilated vascular loops. So this would support their involvement, morphologically support their involvement in immune cell recruitment. Do these cells have any interaction with each other? Well, again, we can make predictions of knowing what ligands and receptors these cells express, how they could be interacting with each other, as they're both found in the dermis. So using cell phone DB to make these predictions, there's a highly conserved, a highly predicted interaction between ACKL1 on vascular endothelium 3 and XCL8 on MAC2. We can stain for these markers, or we can stain for these cells in skin and find that they are indeed CD31 as a. We find that they are closely opposed to one another in tissue. So to summarize, we find developmental cell states and gene programs re-emerge in inflammatory states. And in particular, we find a population of vascular endothelium that we call VE3 that supports immune recruitment, and a population of macrophages that we've called MAC2 that support vasculogenesis. As I said, this was the work of a large group of people who I won't mention all the names, but acknowledge here. Thank you very much.
Thank you very much, Gary, and also thanks to Sally for these excellent presentations. So we will do the questions for both of you together. I'm sure we cannot address everything, so you will hopefully be available for additional questions and answers via email. But let me begin with the first questions that are in the chat and in the Q&A. So a question to Sally is, given the loss of alpha-5 integrin in one knockout model, is there an effect on the expression of fibronectin and basement membrane components? Have you investigated that?
Yes, so indeed in some of our knockouts we have effect of both on both the laminins and fibronectin and deposition by the keratinocytes. So, but we haven't checked in this specific knockout, which is the MGAT1 knockout without complex and glycans, if there is an effect on fibronectin. But we will do that.
Another related question, is there an effect on the Wnt signaling if you knock out glycans?
Yes. Short answer. We are onto it.
Okay, and let me ask another question. So is the glycosylation differentially regulated during the keratinocyte differentiation? So is glycosylation different in the uppermost layers from the lowermost layers?
Yes, yes it is, and that's very interesting. Thank you for the question. It's a very interesting thing because, for instance, with the O-glycans, we have a change from the basal cell layer from a specific structure which is called silyl T to T right after the basal cell layer. So it is regulated throughout the epithelia, and we have expression of different glycosyl transferases in the different layers. Some are all the way, and some are only in specific layers. Yeah.
Thank you. A question to Gary. You have used a relatively small number of samples. Can you say anything about the heterogeneity among the psoriasis samples or among the developing skin samples, or is the number too low?
Yeah, so it's a problem, this approach, although I think increasingly people will be able to use more and more donors as the technologies expand. There is inevitably some heterogeneity, which is how we've tried to address it by looking at the bulk data. But inevitably with these few donors, there is a lot of inter-donor variation. So there's, yeah.
Okay. Another question about the vascular genesis program. Could this be a target for the treatment of or modification of psoriasis or atopic dermatitis? Have you investigated that already?
Yeah, so not specifically, but because it's seemingly inactive in healthy adult skin and then reactivates, the theory being that it's active in development, it becomes inactive and then reactivates again. It's just finding a target that's that would be suitable, that isn't going to be active in healthy skin, which I think is challenging.
Okay, thank you. I think because we are already a little bit over the time, maybe we can ask the viewers to address you personally by email. And I want to, well, let me see, there is one more question, so we have time for this one. The MEK2 with anti-inflammatory properties are upregulated in inflammatory skin diseases. How could you explain this?
Great question. It's what we've been talking about this a lot ourselves. So whether or not it's to try to act as a sort of negative regulator of inflammation, we're really not clear on that at all, but we are looking at it.
Okay. Okay, I guess many new data that needs to be followed up in both studies. So thank you very much, Gary and Sally, and I hand over to Edel, who will give an outlook to the next kitchen presentation.
So thank you so much, Gary and Sally, for joining the ESDR Kitchen. We very much enjoyed your talks. So the next episode is going to be on the 24th of February at 13:30 CET. And it's going to be Masa Amagai, who will be talking about his career and his work on pemphigus. So I'm sure that that will be very interesting and very inspiring, and we look forward to seeing you all then. Thanks.